So yes, I'll start over. My name is Lauren Cheraboga. I am one of the co-presidents of the Society of Physics Students, and this is one of the first undergrad sponsored colloquium here, where we all voted to invite our lovely guest, Dr. Vijay Balasubramanian, to come speak to us about a very cool topic, imagining space. Dr. Balasubramanian has a PhD in theoretical physics from Princeton and is currently doing work at UPenn as a professor of physics in such a variety of topics, all ranging from understanding systems in the brain to string theory, which how that can all stay straight is a mystery to me, but it's very exciting. And yeah, without further ado, to present on imagining space, Dr. Vijay Balasubramanian.
So thanks very much for inviting me. It's a real honor always to be invited by students as opposed to somebody else, because I always feel like that's really special. So thank you so much for inviting me. And I really enjoyed the conversation I had with the physics students during the previous hour. Very interesting and very smart people. So thanks a lot. Great. So today's talk is entitled Imagining Space. And so let me start off with a question of what is space in the first place? So, since ancient times, people have understood space as some kind of stage on which physical things are located and on which they move and interact. And so if you go around reading the cultures and traditions of peoples from the beginning of the recordings of these ideas, and even in cave paintings and so on, people seem to have thought about space as variously as infinite, as finite, as flat, as round, as continuous, even sometimes discrete in various ways, centered on Earth, centered on the sun, centered nowhere, and so on. So many, many, many different conceptions of what space is and what the relationships between different places in space can be. Space can be real, as in the three-dimensional space we talk about, or can be abstract. So for example, people have thought about the cosmos as a whole as a space. Here's a picture on the right of such a picture of the cosmos as a whole. These are mandalas, which are things that pictures drawn in India, Nepal, Tibet, places like that, which are supposed to be cosmic diagrams of the kind of geometry of space. I don't know how to read this geometry, but it's supposed to somehow be a map of the relationships between all the places and entities of the universe, right? I don't know how to read it. So, of course, some notable things about space is you can move in it, and you can move between spaces. Even this has a whole variety of different conceptions. So, for example, in Mesoamerica and Australia, traditionally there were always 4 cardinal directions: north, south, east, and west in our language. But in India, there were 10. There was north, south, east, and west, and all the intercardinal directions in between them, and up and down, right? And they have names, for example. So many different conceptions have existed in this way of space. Now, in modern language, in the way we talk about space in physics, it's much more stylized. So really, our modern notion of space as we use it in physics really got solidified, I would say, by the work of Descartes, who lived between 1596 and 1650. And he really introduced the way of thinking where we think of space as a stage for action described in a coordinate system with some number of dimensions, and you quantify where you are, and then you quantify motion that's going from these coordinates to that coordinates. And most of what we do in dynamics and physics is described that way. As we all know, a few centuries later, Einstein taught us that this picture is sort of misleading a little bit. Really, space is perceived relative to an observer's motion, and space and time can be mixed in space-time, and matter warps space-time, like in this image from the LIGO collaboration. It's even worse than that, or more complicated than that. Very recently, those of us who are string theorists have even learned that space can be emergent, right? It may not even be there. It can just be a convenient way of describing a physical system that doesn't even have that space. It's just sort of an effective thing. You put it down because it conveniently summarizes in some effective way the dynamics. So even that is possible. So these are many conceptions of space that societies have had, and that as we've had them in physics. But part of the reason we spend so much time talking about space and place is because we're animals. An animal life is defined by motion in space. The word animal almost means motion, to animate, right? The notion of anima or soul is associated with motion, right? There are other kinds of life, and for example, plants. And if we stayed all the time rooted in one place, we would be plants, and we would have to have that sort of lifestyle. But we're animals. We move in space, and therefore we think in space. There's a great deal of your mind that's built around space and how to think about it. So, for example, if you're an animal, you have to take actions. You got to move in space. And because of this, because there are lots of spaces and they're different from each other, you have to have within your head the capacity to learn the geometry of new spaces, to imagine where you are in them, and how to get elsewhere in them. And it may be all of you, or maybe just your arm and things like this, but you need to have the circuits and capacities to tell you about these kinds of spaces. Very often, animals like humans anyway, and perhaps other animals too, it's hard to query them, also have in their head the geometry of abstract spaces. We geometrize everything. We talk about relationships. We say we're close to somebody else to mean that we have a good relationship with them, or we're distant from that, or I feel distant from that idea to say that we don't agree very well with it. Right? So we use space everywhere in our mode of organizing thoughts. So, but inside your head, what is space and place? So inside your head, here, this location, this is my living room, for example, is sort of an abstract pattern of neurons firing. There is no space, there's no Cartesian grid sitting around anywhere directly in your head. There's no picture that you can draw on. It's just neurons that are firing in some pattern. And these patterns somehow cohere to construct what's called a cognitive map of your location in physical spaces, and as we're recently starting to understand, even in abstract spaces that your mind navigates. The same structures are used for both of these things. So in this sense, space in your head is always an emergent quantity, right? It's not really there. It's kind of an abstraction that describes interrelated patterns of activity, sparks of electrical activity that happen in your brain. So the purpose of my colloquium today is to tell you about some of the recent discoveries that have happened on how this that have occurred on how this happens inside your head. One of the reasons why I'm very happy to be giving this colloquium is the last time I gave a colloquium at NYU, which was more than a decade ago, it was about how space was emergent in the sense of string theory. So I'm delighted to be giving a colloquium about how space is emergent in the sense of neuroscience today. Okay, so let's discuss how would we think about how this happens. Well, to think about that, the first thing we need to know something about is what the brain is made of. Because if we're going to talk about how neurons in the brain conspire to produce maps of the world and cognitive maps and so on, we need to know what neurons are and how they're hooked together. So since this is a physics audience, and many of you may not have that background, I'm going to spend a few minutes introducing that. So let's start by discussing circuits in the brain, what they're built out of and how they work, so that you have some background about what things I'm going to say later. So first of all, the neurons, your brain is made of neurons, and the neurons are cells like any other cell in your body. They have cell bodies, they've got cell membranes, they've got all these kinds of things. But of course, the way biology works is the different components that make up living things are adapted to the functions they have to perform. The job of a neuron is to compute and communicate. As a result, it has these input wires on one side called dendrites, and at the input wires, there are synapses that connect to other neurons, and signals from everywhere else are gathered in these dendrites and come in as little electrical pulses, currents that go into the neuron. At the other end, there's the axon, that's the output wire of the neuron, and the axon ends again in a bunch of synapses where messages that are sent down the axon are communicated to the next neuron. And these things are somehow going to conspire to create your sense of space and map of the world. Now, how do neurons communicate? Well, they do it through electricity. They have a baseline potential, about minus 70 millivolts. And when you stick in current into them, the potential sort of increases. It bounces around kind of noisily and kind of increases. And then there's a runaway feedback process by which if it reaches a certain threshold, it goes boom and fires and what's called an action potential or spike. The voltage sharply goes up. And then there's another runaway feedback process that sharply brings this back down within a millisecond, and then goes back down and comes back to baseline. So in this sense, it's electricity that makes neurons function. And really, everything in your head, it's a computer of a certain kind with electrical connections being produced by these kinds of neurons, sending all the messages back and forth. In fact, in the early days of the discovery of electricity, when people like Galvani and Volta and so on and so forth were doing their experiments, people discovered that the connection somehow between electricity and they didn't know there were neurons then, but animal motion, because they, what people did is they would take things like dead frog's legs and stick an electrical pulse. The early batteries had just come to be, so you could just take the battery and stick it around the frog's leg, and the frog's leg would kick. There were crazy experiments done where people would take executed criminals and pump them full of electricity, and then the body would go like this because, they didn't realize this. They thought that meant that life was basically driven by electricity, but it wasn't. It was just that neurons, all that power the muscles, were being fired by the electricity, so the body would begin to move. And indeed, this led to all kinds of things, like the story of Frankenstein by Mary Shelley and so on, ultimately had their roots in the fact that neurons are driven by electricity. Well, but single neurons like this aren't going to express love, write poetry, or indeed produce your map of space. So you need many neurons working together. So how do they work together? So let's go to the other scale, the whole brain. And if you look at the entire brain, the brain is divided up into different kinds of areas where there are circuits that do different things. So for example, over here in the back of your head are the circuits that are responsible. This is the visual cortex. They're the circuits responsible for vision. This is why if you fall back and bash the back of your head against the ground, you have blurry vision for a while. That's because you've got kind of a bruise on these circuits and they're not functioning right. And so you can't see quite straight. Right? It's as simple as that. This is why in this region that does motor control, if you have a stroke, then a piece of your face will sag, for example. It's because there are circuits here that are responsible for doing a particular thing. This has also been known for a very long time, that there is something like this going on. There's a papyrus from some building site and some doctors in Egypt from a way long ago, many thousands of years ago, in which the author of the manuscript says that, he's noticed that if somebody has damage in this part of the brain, then one thing stops working, but everything else works, and things of this kind. This kind of localization of function was discovered very early on in human history. And in modern times, it was really brought to the fore, but in the 19th century, for example, there's a Rue Broca in Paris, which is named after Dr. Broca, who had a patient who had damage to a particular area of the brain called Broca's area. It's right here. And if you have damage to that part of the brain, everything else will work fine, but you can't produce speech. And so that's at that level that things are sort of focused and that the circuits are specialized. If you look even a little bit closer, you take one of these areas, like the area that does motor control, you'll see that the circuits on this side of your head control your face, and over here, circuits that control your wrist and so on. So the circuits are carefully organized. And that's, of course, very helpful to us because we're interested in the circuits that control your spatial cognition. So in the same sense, there is an area inside your head there called the hippocampus. And the formation around it, the general hippocampus formation, it consists of various areas that we'll talk about, which work together to build your maps of space and to give you your sense of where you are in the world. Okay, so we actually need to go one more step down because it's important to realize that that's not the whole, that at the more microscopic level, there's also specialization. This is because when I start talking about the circuits in these different parts of the brain, I'm going to start talking about particular kinds of neurons of one kind or the other. So if you take a particular area of the brain, here this is a picture of the retina drawn by Ramon y Cajal in 1917, you'll find that if you look at this picture, right, he drew what he saw under the microscope. This looks like a 3-layer neural network. And the first layer, there are the photoreceptors, the rods and the cones. Then there are the so-called bipolar cells. And then there are the so-called ganglion cells, which are the output layer of the retina with the wires coming out forming the optic nerve. So there's this sort of intricate sort of microstructure to the circuit with many types of cells cooperating to do the function, just like in an integrated circuit in silicon. So now using modern techniques, molecular techniques, electron microscopy and so on, we know that this repertoire of circuit elements that was drawn by Cajal was a bit coarse. And in fact, there are more than 60 different types of circuit elements in retina.
Here's just a little correction. Is Ramon y Cajal is actually all is the family name, is the last name.
What's that?
Ramoni Cajal is just the last name of the person. So it was like, so wasn't Ramon first name and Cajal last name? Ramoni Cajal was the last name.
Fair enough. Okay, great. Ramoni Cajal is the entire last name, and his first name was?
Last name, I don't remember, but it was.
So I have never heard anybody called Ramoni Cajal anything other than Ramoni Cajal. So I will take this as a revelation and go dig out.
He had a name, yeah, he had a name.
I've never seen any other name mentioned. Amazing. Camilo Golgi, who was his competitor, who was clearly Camilo.
It's
That was the
Santiago.
wrong one, yeah.
Santiago Ramoni Cajal. Yes, that's
Santiago
it.
Ramoni Cajal. Yeah.
Who said that? Whoever said that. That's it. Santiago Ramoni. So it turns out I had heard the first name.
But it's confusing because in biology, right, there is a Cajal body.
Oops, sorry. Yeah, yes, there is.
Exactly. So they shortened his name already there. The last name.
It must be because of all these people who are not Spanish didn't realize Ramoni Cajal was talking to the Cajal. Well, I stand corrected. There you go, Santiago Ramoni Cajal. So I should have put here Ramoni Cajal. Okay, Ramoni Cajal, 1917. But what I was trying to say, what I was trying to go after, was that there are many different types of cells. So in the circuit, for example, there are 65 of them. They're all shaped differently. And usually in biology, form follows function. So all of these different kinds of circuit elements do different things. So for example, these amacrine cells are responsible for sort of filtering out information from the visual stream and kind of shaping the input that goes to the central brain. And altogether, they cooperate to create your perception of the world. So in a similar manner, for our purposes, we are interested in the circuits that underlie our sense of space. So if you think about those, and think about all of these different layers of organization at the same time, here is a sort of picture of a slice through the brain showing the relevant piece of the brain. And here is a diagram of the many different areas of the brain that cooperate together in order to create your sense of space. So one thing that you're going to need is external sensory input, right? You're going to need to get, your sense impressions of the world, your visual impressions, and other kinds of things. And that comes in through two areas called the perirhinal and postrhinal cortex. Then you're going to need something, if you want to know where you are in the world, you need to know something about how you're moving, right? Either because you can feel yourself moving or because you can see yourself moving. And that movement comes into the circuit through a different area. So different areas called the pre and parasubiculum and something called the retrosplenial cortex. The names don't matter much for us. All of this information is then integrated in two areas of the brain that will primarily interest us, called the entorhinal cortex and the hippocampus. And in those areas, there are circuits and cells that are charged with maintaining representations of where you are in the world and making plans for how to go, helping you to make plans for how to go from one place to the other place. And just like in the retina, we saw a zoo of pictures of different kinds of cell types. Within those areas, there is a zoo of types of cells that will cooperate and that we will use a little bit later. There are border cells that sort of tell you when you are in a certain piece of the boundary of the space. There are boundary vector cells that tell you that that there is a boundary at a certain vector direction, in a certain vector direction. The head direction cells that seem to be tuned to which direction your head is pointing in the world. There are speed cells that are tuned to how fast you're moving. There are place cells that are associated with the sort of spatial context and other contexts of your behavior. Landmark cells, object vector cells, and I left out the most important one that I want to talk about today, called grid cells, which are somehow basically fundamental to the creation of your spatial map. So my goal today is to tell you a little bit about what we've learned about understanding the logic of these circuits and how they produce our spatial imagination. So the approach we're going to take is, okay, so I need to understand a theory. One would like to have a theory of these kinds of circuit repertoires. So we're going to think about that circuit repertoire as a kind of memory of the computations that have predictive value for behavior that were learned over evolutionary time and are encoded in the genome and the developmental program, right? So in other words, I'm sort of interested in understanding what are the organizational principles or laws, if you like, that control the computational and information processing repertoires of the brain. And the basic idea is that these repertoires have been selected because they do these kinds of things well, namely they help you predict what you need to predict about the world well. So there's a nice book that I'd like to recommend by Peter Sterling and Simon Laughlin that sort of sets out this kind of approach to thinking about circuits in the brain very nicely. So you might enjoy that. It's called Principles of Neural Design. So from this body of work in neuroscience, today we're going to use two principles. So if you want to understand a circuit, two useful things to think about are, for example, well, we're going to start out by talking about the costs of computation that will lead us to the two principles. So some of the principles that people talk about in the organization of neural circuits arise because neural circuits are expensive to maintain. So your brain is 2% of body weight, but it's 20% of metabolic load. What's more, in terms of space, every millimeter cubed of the brain contains about 4 kilometers of wire. At the same time, the brain only consumes 12 watts of power, which is about a refrigerator light bulb, much less than my laptop, and the packing seems to minimize wire length. So there's been a lot of thinking in theoretical neuroscience and experimental neuroscience on how the brain manages to achieve this efficiency. And by thinking about that, two principles that people talk about in the organization of neural circuits is one, is that circuits should be adapted to the structure of the world so that they invest their limited resources well to achieve whatever function they need to have. And the second principle people talk about is that once you have the basic structure of the circuit, it needs to be able to self-organize and learn to adapt to changes in the structure of the world. So we're going to talk about both of those aspects of our circuits today. So just to emphasize that, the first idea is that circuits in the brain efficiently use resources. So the idea is that the brain exploits the average structure of the world that they must interact with to efficiently allocate limited computational resources to maximize gain for the organism. So we're going to take this attitude to thinking about the entorhinal cortex and how it might be organized to represent space. So that, roughly speaking, the idea is, suppose this rectangle is a cognitive function or a sensory feature space or something that needs to be covered, then you imagine the different architectural elements, circuit elements, if you like, that you can use to cover this space, and that you somehow need to tile this well. You don't want to over-represent some regions of the space or under-represent other regions of the space. You should cover it well to do your function. The second principle, as we just said, is the idea of learning and self-organization. So once you fix the basic structure of the circuit, the actual worlds you encounter, vary from the average structure that you're adapted to. So it's necessary for neural circuits to self-organize their architecture through dynamics and learning in order to adapt to these variations and to learn new task environments. So my talk is going to be basically divided into two parts. One in which I will use this idea of adaptation of circuits to the structure of the problem in the world to predict and understand various things about spatial cognition. And then I'm going to talk about how these structures can wind up being produced by self-organizational dynamics. Okay, so that's what we're going to try to do. So I'm going to start with the first part, which is thinking about imagining space inside your head. So here's my living room again, and we want to know something about place and space, and we'd like to know how abstract patterns of neurons firing in your brain might be able to maintain a map of your location. So it's helpful in doing this to start by just forgetting about the brain and asking yourself, if you were going to design a system to represent space or place, what would you do? So here's a simple world. It's an 8-meter linear track. And let's suppose that for some reason, you needed to have 1 meter resolution in the for space in this world. For whatever reason, you need to have 1 meter resolution. One way of producing a representation inside your head that would have that resolution is to divide this 8-meter track into 1 meter blocks. And then imagine that you have a neuron that responds if you're in this block or this block or this block. So for example, if my cursor is a mouse and and I'm talking about neuron 3, as I move my mouse, I might hear brrr, and so on, right? So in other words, this neuron 3 would fire when the mouse, the actual mouse, right, the rat, is in this location in the world. So this is a scheme for representing place in the world. You just lay it out in a linear map, and you have assigned a neuron to each place. Notice that this requires 8 neurons to represent this 8-meter world. So in fact, in the hippocampus, there are cells that do something like this. They're called place cells. But when you examine them in more detail, place cells don't just seem to encode space. They encode smells and sounds and abstract things and objects and all kinds of stuff at the same time. So they seem to be more like cells that encode some sort of conjunctive context for behavior. So they're not exactly about representing space. It's sort of putting all kinds of context together. So just that's a comment in passing. So now I want to think about another representation of that 8-meter world. So imagine that I took this 8-meter room and I divided it into two parts, the left part and the right part, right? And I called the left part 0 and the right part 1. And then I subdivide again recursively into, I take the left part and divide it into left and right, and the right part and divide into left and right, and call those at a second scale. I'm going to call this the 0 neuron. And the 0 neuron may fire if the animal is here in the first blue patch or in the second blue patch. And the 1 neuron may fire if it's in this patch, if the animal is in the first orange patch or in the second orange patch, and so on. Right? So we do this recursively. So in this representation of space, if the animal is at this dashed location, you might call that the 0, 1, 0 location. And this dashed line is the 1, 0, 1 location. In other words, this is a binary code for space. Notice that it's slightly more efficient. It only requires 6 neurons, which do this kind of recursive subdivision to represent space. You could, of course, use other bases if you felt like decimal. Okay. Now, we're interested in representing two-dimensional space. So suppose we think about that. So here's a square. Let's suppose it's 8 meters by 8 meters. And let's suppose I want to have 1 meter resolution, 1 meter by 1 meter resolution. Then if I was to have a place cell scheme in which I encode location by dedicating neurons to each location in the space, and then lay out the space in that way, neuron fires, I know where I am, right? Then you would need 64 neurons to describe this world. Now let's consider the two-dimensional analog of binary numbers. So I'm going to divide the square here into 4 parts, green, yellow, orange, blue. Then I'll take each of these squares, subsquares, and divide them into 4 sub-pieces. And then I'll take each of the little pieces and then subdivide them again to 4s. And now, for example, the location marked by the cross is the blue, yellow, blue location, right? So here you should be thinking about each color as representing a neuron. So the blue neuron at this scale, at the biggest scale, fires in the top left corner. The blue neuron at the intermediate scale fires in this grid of locations marked by the blue, and so on. Right? Very bizarre sounding scheme in some ways, but notice it's much more efficient. You only need 12 neurons to represent an 8 by 8 grid world with 1 meter resolution. Okay, so you can use other bases too, like decimal if you wanted. Now, preposterously, perhaps, turns out that there are cells in a region of the brain called the entorhinal cortex that behave just like this. So in the entorhinal cortex, there are things called grid cells, and they respond when an animal is physically in locations lying on a triangular lattice. So for example, at this scale, you may find a grid cell. So this is at a given location in the entorhinal cortex. You might find a cell that responds on this triangular lattice of locations. By the way, this is data, right? There's an animal wandering around this square room, and this heat map indicates where the animal was when the cell responded. So there's this grid of locations. If you look at neighboring cells to this one, you'll find that they have randomly varying offsets or phases for their grids. Then, if you go down the entorhinal cortex, actually from the dorsal end to the ventral end, from the back end to the stomach end, you'll find grids of increasing sizes. What's more, the grids that appear in this way are organized in discrete modules. So there's one scale, and then it jumps to another scale, and then jumps to another scale, and each of these modules of a given scale share a period and an orientation. So looking at that, I would say that this picture on the left looks just like the picture on the right, at least it's on qualitative level. So it suggests that what the brain, the entorhinal cortex, is implementing is kind of a two-dimensional fuzzy neural number system for location in a two-dimensional world, for navigation in a world. So you could ask, how could you get some evidence for this? Well, so suppose you agree that grid cells, which were discovered 2005 or so, are a numbering system for location. Well, you might say, well, if I have a broad principle that says that neural circuits should be efficient, then presumably they should pick a number system to represent space that's efficient in the sense that requires the fewest resources to give me the resolution that I require to behave in the world. So that would seem like a reasonable hypothesis given the rest of the experience we have with sort of efficient coding and efficient computation in the brain. Okay. So if you ask the question, what number system should you pick? Binary, decimal, etc., right? That could be a way of asking that question. Well, in this language of grid cells, the ratio of the grid periods between adjacent grids is the base of the number system. So for example, if you went down the entorhinal cortex and you found that the grid size, the period multiplied by 2, there was a geometric series of periods multiplying by 2, then you would say it would be like a binary system. But of course, these are neurons, and there's no actual requirement that they write the numbers down on a piece of paper. So there's no requirement that the base be an integer. This could be an abstract number system with a non-integer base, for all we know, right? Okay, so the way we're going to test the hypothesis that this thing that you see in the brain should be interpreted as a kind of exotic number system for location in 2 dimensions is by saying that it ought to be efficient according to other principles in neural coding. So we're going to try to predict what the most efficient base is going to be. And we're going to then compare that with the data. So for simplicity, let's do this in 1 dimension. All right, so imagine you have 1-dimensional grids. So this is the picture that, at the first scale, you have, the red cell responds on the left side of the room and the blue cell responds on the right side of the room. So the period is like lambda 1 for the grid that you're making. Then at the second scale, you have the red cell respond to the left half of the left half and the left half of the right half, like this. So there's a second period and a third period. And so you're going to combine the information from cells responding to these different regions to figure out where the animal is. So more realistically, if you're working with real neurons actually, they don't fire in little square firing fields like this. Rather, they'll fire most in the middle and they'll tail off a little bit at the end and things of this nature. So we'll discuss that separately. Let's think about the idealized situation instead, because it's easier to think about. Now, clearly, in order to get resolution, in order to get the fine resolution you need about where you are, you would like to squeeze these periodic grids down as much as possible. So as you squeeze them down, right, the resolution you get is really given by the size of the smallest grid. But notice that if you squeeze it down too quickly, ambiguities will appear. So if you get greedy and you go from this scale and you rapidly squeeze down to the next scale in order to get fine resolution, you'll find that at this second scale, there are sort of 2 red regions underneath this red region. So the red neuron fires at the bigger scale and the red neuron fires at the smaller scale. You won't know whether the animal is here or whether the animal won't know if it's here or here. So there's a constraint here that says that the size of the firing region at one scale, scale I, should be bigger than the period at the next scale in order to have unambiguous encoding. Okay? So with that information in mind, we're now going to try to build the optimal grid and then compare that with data. This is for encoding space. So I'm going to draw the pictures in 2 dimensions, but I'm going to write the equations in 1 dimensions, and I will tell you how to do for any d dimensions that you want. So in the entorhinal cortex, so here I'm in full theorist mode, I'm building a model of the entorhinal cortex. The entorhinal cortex is like a strip. There's the dorsal end near the back of the animal. There's the ventral end at the other side. As you go from the dorsal to the ventral end, you find cells, individual cells that fire on grids in the world that are of increasing sizes. Let's parameterize the period of those grids by lambda I and the size of the firing region by L sub I. Then a parameter for how this thing scales up, how it telescopes, is the ratio of grid scales. The period at one scale divided by the period at the next scale tells you how quickly the thing telescopes. And the resolution of this whole system, which tells you how many pieces you can divide the room into, is the ratio of the biggest scale to the smallest scale, which is equivalently a product of all of these ratios between adjacent scales. So the animal has some behavior that it desires, And so because of that behavior, presumably it divides up, it needs to be able to divide up the room at a certain resolution. So this resolution we're going to keep fixed as given to you by the animal's behavior. Clearly a human versus a mouse needs to divide up space into different sizes. Humans can tolerate more ambiguity in their location because their feet won't fit in a smaller region anyway, right? That kind of thing. Right. Now let's talk about the cost of this system, right? So in order to create such a grid system to encode space, what do I need? At each scale, if I make one of these periodic grids, I need to have enough phases for the grid to cover space, right? Because there's a cell with this phase, then I need to make a cell with that phase in order to cover space completely. So the number of phases you need to have is the period divided by the length, by the size of the firing field. Well, in d dimensions, this would be this thing raised to the power d. So in 2 dimensions, it's really squared because it's the ratio of areas. But you see what I'm saying. And in this way, by summing over these different scales, by these different modules, if you like, you find the number of grid cells you need. Okay? So we're going to assume, fairly, that the cost of running the grid system is some monotonically increasing function of the number of cells. That makes sense from many points of view. Every cell costs you ATP to run, it has connections, it does this, it does that, and all kinds of things. So I don't know what the cost is, hard to determine in detail, but it's certainly going to increase with the number of cells. So that suggests that what we should do to get the minimum, to pay the minimum cost for getting this resolution is we fix the resolution and we design a grid system that gives us the desired resolution for a minimum, with a minimum number of cells. We also have a constraint for unambiguous decoding. We already said what that was. The period at the next scale must be, which is equal to the period at the previous scale divided by these scale ratios, must exceed the width of the firing field at the previous scale. This is a constraint we already wrote down. And that tells us that these numbers, lambda I over Li, the period divided by the width of these regions, must exceed the scale ratios, right? So because it must exceed it, and since the number of cells is proportional to these lambda I over Li, to minimize n, the number of cells, the cost of the system, you might as well just replace everything on the right-hand side by these Ri's, by the scale ratios, right? Because that's the smallest they can be for unambiguous decoding. So that leaves us with the following problem. We parameterize the system in terms of the ratio of the scales of the adjacent modules. The resolution that we have to keep fixed is a product of these scale ratios. And the cost of the system is a monotonically increasing function of the number of cells, which is a sum, or is proportional to a sum of these scale ratios. So now we have our problem. Take a bunch of numbers Ri, fix their product, and minimize their sum. Easy. You can solve the problem easily in about 3 lines of math, and you discover several interesting things instantly. First is you predict that the ratios of all the adjacent periods must be equal. So basically, the ratio of period 1 to period 2, period 2 to period 3, etc. must be the same. So that's a prediction from this theory. The second prediction is that this ratio that you should get if you want to have the most efficient grid system for location is that this ratio should be the dth root of E in d dimensions. So in 2 dimensions, that should be the square root of E. So when Shui-Shin Wei, my student myself, worked this out on the blackboard, I remember telling him, oh, that is so beautiful, but it's going to be wrong as far as biology goes because there's nothing in biology that ever works out so neatly, right? Just you wait and see. Well, to be completely serious about applying this to the real physical system, to the real biological system, we have to be a little bit more careful. We have to understand that real neurons are noisy, so when they respond, what you actually know is something about, if you do Bayesian decoding, you will know that the location of the animal is somewhere in this black region, let's say, with some probability distribution. When you go to the next scale, you'll know you're either here or here or here, at a finer scale. And then really what you should do is you should multiply these things together to get shrinkage in this probability distribution. You can get ambiguities if these things shrink too quickly, and so on and so forth. So there's a little bit more work to do than the cartoon way I did this for the purpose of this colloquium. And really, you're really in 2 dimensions, so there's some doubly periodic lattice, and you've got to minimize over that and do some more work there. Let's avoid that and just tell you what the answer turns out to be. So it turns out that if you take the simple model that I just talked about, the answer is square root of E is what you should have as this adjacent scale ratio and should be constant. The probabilistic model gives slightly different answers. The answer actually, strictly speaking, depends a little bit on exactly some details of the assumptions. And so I've put a theory error bar here because there's some plausible range of assumptions you can take and so on. Okay. Now here are two data sets. This contains hundreds and hundreds and hundreds of cells. And there's another data set, again, that's come out recently with even more hundreds and hundreds of cells, all squarely and exactly where the theory predicts it to be. So this is an excellent match with the theory, right? So in other words, if you were inclined to be poetic, you would say that evolution seems to have invented a base n number system and then optimized it for neural hardware. Right? And it isn't base n as an integer, it's base square root e. Now, how cool is that? So a very nice thing here is we seem to have explained the organization of a rather complex circuit supporting a cognitive function from an efficiency principle. We seem to have explained that it gives an efficient code for space. Okay. So, as I said, actually, it's not enough just to say you have this efficient code for space because, you go into new spaces all the time. And so presumably, you need to make maps in new spaces, and every new space you go, this thing has to organize itself to produce this kind of scale ratio and efficient code and so on. So in fact, let me justify to you from the experiments that indeed these grid systems have to self-organize. They're not sort of wired in from the beginning. So it turns out that when an animal explores a new environment, initially these grid patterns don't occur in its brain. It wanders around for about half an hour, and at the end of half an hour, there's a well-formed grid pattern suggesting that there's a learned self-organized map. You can say more. Suppose you put a Q on the wall, like a red dot, and it learns the map, and then you turn the Q, right? Then the whole map rotates. It's as if when you learn the map, there's some anchoring that goes on with sort of the faraway landmarks, and then you anchor it that way. And as you move the landmark, the map turns. So it's clearly not sort of a static thing that's wired once and for all. What's more, it's clearly not just based on visual input or sensory input. You're using your self-motion. The way you know this is the grid's persistent darkness, right? You don't have to be able to see anything, and the grid will persist. So it's clearly coming partly through path integration, right? So you internally to your body, you have access to the speeds at which you're moving your various muscles and the way you're turning your head and this, that, and the other. So clearly the body can do that to create and maintain these maps. Okay. So we're going to use this to consider a dynamical theory of self-organization that can produce this kind of a hierarchical map, that an efficient map of location. So to do that, we have to treat neurons as dynamical systems. Here we have Ramon Cajal, now correctly done with his last name. Let me tell you, for the people who are not do not do neuroscience for a living, the way you think about neurons as dynamical systems. So the most naive way of doing this goes back to a person named Lepic in 1907, same year as special relativity, actually, in which you think of the neuron as a little box. You stick in some current. The current is linearly integrated up, but there's a little bit of leakage of charge. And so then what happens is that then you say that when this integration reaches some threshold, you declare by hand that the neuron has fired and bring it back to baseline. You just do it by hand. Okay? So that you can certainly implement in a computer, or you can do some calculations by hand. That's one level of neural model. Another level of modeling as a dynamical system is something called the Hodgkin-Huxley model. This won the Nobel Prize in 1963. And this is a way of modeling which is completely realistic. You model all the stochastic processes and the biophysics of the voltage-dependent opening and closing of ion channels, the whole bit. Huge effort. It gives you the detailed shape and dynamics of all the voltage spikes. Beautiful thing, but probably overkill for many purposes. So there's an intermediate thing that came out about 10 years ago. I love this model. It's called the Izikevich model, and it's an effective theory in the full sense that we would talk about in physics, right? So Izikevich went and looked at these Hodgkin-Huxley models, and he fiddled the parameters, and he found out these are dynamical systems with attractors and bifurcation points and everything, understood it, and said, I can make an effective model with 5 parameters that will accurately summarize the coarse-grained dynamics. And depending upon how you fix the parameters, you get things that spike regularly, that burst, that chatter, all the different kinds of dynamical phenomena that you expect to see from neurons. So for what we're doing today, we'll use the leaky integrate and fire model, but you can use any of these things because we're going to be talking about the universal dynamics, self-organizing dynamics of these systems, and you can do whichever of these you like, and you would get that. So I just wanted to introduce the idea of a neuron as a dynamical system. So how would you self-organize these kinds of grids we've talked about? So at the first level, I don't want to think about all the different modules. Let's just think about one module, so one scale at its period. So there's a standard model for this due to Yoram Burak and Ilafit, and it goes like this. You first imagine a sheet of neurons. This is supposed to be the entorhinal cortex or something, and there's a sheet of neurons. And there's a global excitation, some drive. It's like an external magnetic field. Something's driving these things, driving persistent activity of every neuron on the sheet. And you describe this, let's say, using a leaky integrate and fire model, the cartoon model where, there's an external drive, that's this A of R, and then there's a decay, that's this S of R, there's some time scale for the decay, and sort of this goes, so there's a maintained firing. Now suppose you add to it local inhibition. So a little bit like an antiferromagnetic model or something, you agree that each neuron inhibits its neighbors with a strength that decays with distance. So it's clear then, if you do that, that if the central neuron here fires, it's going to shut the ones next to it down. And then it'll release the one farther away, and that can fire, and so on and so forth. So it turns out that if you write down a dynamical equation for this, it's driven to a stable attractor. And the stable attractor is a triangular firing pattern of neurons on the sheet. So this one will fire, then this one, then this one. There's a triangular pattern of neurons firing on the sheet. Okay. So notice that this pattern breaks the translational symmetry that was originally present in the system. So that means, of course, you could have shifted this pattern in any way, and it's still going to be an attractor pattern. Right? The initial conditions will tell you which of these things you get driven to. So that means there's going to be a continuous manifold now of phase-shifted attractor states and so-called zero-energy modes that allow you to translate on that manifold. So now comes the really clever trick. So what you do is you couple these zero modes, these easy translations in this dynamical pattern of activity, to the motion of the animal in the world. So you imagine there's another signal coming in that is the velocity of the animal. And it's known that in this area of the brain, there are such velocity signals derived from the self-motion or from optic flow. So by the signals from within the body or from optic flow. So you couple those together. And so if you do that, the coupling basically moves the pattern on the neural sheet as the animals move in the world. So if you want to be mathy about it, you might say there's an isomorphism between the neural sheet and the world. The neural sheet becomes a mirror of the world. And as the animal moves around, the activity pattern translates in cortex. So this has a really cool outcome, right? A given neuron that on this neural sheet fires then when the animal occupies a lattice of locations in the room, exactly as the doctor ordered. And these days, there's mounting experimental evidence for this kind of thing. You can image these cells, you can work out the anatomy, you can work out detailed checks of what this kind of attractor dynamics would do. And there's mounting experimental evidence. This kind of model probably describes what's going on in the head. But for today's purpose, since I emphasize so much the different modules and how they cooperate together to build a map of your location in the world, our interest is how do you get this geometric series of periods? Especially, how do you get the square root of E that you're supposed to get, apparently? Okay. So you might think, well, okay, so I had one module, here's a second module, here's a third module, lots of these modules, going depth-wise in the entorhinal cortex. Well, one way to get modules that have different periods is to increase the inhibition length scale. So suppose I have a neuron firing, and it shuts up neurons in a bigger radius around it, then I'll spread out the pattern, the attractor pattern. So you could definitely do that. Okay, so if you do that, there you go. You add inhibition that pushes the thing out. What you'll find is that the grid period varies continuously with the inhibition scale. No surprise. In each of these layers, if you have a neuron fire, it shuts up things around it with a certain radius set by the inhibition scale. So if you change that continuously, you'll get a continuous series of different periods. But that's not what we want. We want a discrete hierarchy. So the question is, how can you self-organize such a thing? So here we can make an analogy with something with a nice problem in condensed matter physics called the Frenkel-Kontorova model. I even took the picture from Chikin Lubensky's book. And this is a model where you do the following kind of thing, right? You have a potential, and the potential has minima that increase in scale between different layers. So if you put particles in the first layer, they're at a certain scale. And then as you move them down further and further, they want to move apart because of the potential minima. Then you add springs to these things, and they want to pull together. So there's a competition between the push and pull that leads to discrete jumps in the periodicity of where the particles sit. So the push and pull compete, and this happens. So we can be inspired by this and consider that what we'll do is we'll have this continuously increasing scale of inhibition, pushing things apart more and more. That's like the potential pushing things apart. But then we're going to add excitatory couplings between these layers. We're going to imagine that this structure in the brain actually has longitudinal excitatory coupling. So this is going to wind up being a prediction, right, of the theory. For this model to work, it's going to need anatomically the existence of such connections. Okay? So we'll add longitudinal connections. So now we're going to have a push and a pull, right? The inhibition in the circuit, layer by layer, is going to try to push the pattern apart to make it bigger. But the longitudinal connections down this axis are trying to keep things fixed and not let things move apart. So there'll be competition. And indeed, if you work out what happens, you get a whole bunch of discrete modules. This is a complicated figure to read, and I'd just like you to focus on the fact that if you look at this figure here, without the coupling, you get a kind of continuous spread of these blue lines. Those are different periods. And with the coupling, you get 3 discrete periods in this particular example that we worked out. So in other words, you get these discrete jumps in the period. What's more, what controls what the discrete jumps in, the periodicity, is kind of the ratio of how strong the excitation between layers is and the inhibition within layers is. And they produce different kinds of coordinations of the lattices between these different layers. And that's what produces these kinds of discrete jumps. So in particular, this is a zoo of different possibilities that comes about. And I've tried to draw that a little bit better here. As you change the ratio in inhibition length scales between neighboring modules and versus the strength of the excitation, you get different kinds of lattice coordination patterns and different phases and so on and so forth. Okay, so that's all very cool, and it's kind of a 2-dimensional generalization of this Frenkel-Kontorova model which has a devil's staircase of phases, and you see all of that stuff here too. But in its application to the brain, what you really care about are some of these patterns what you actually see, because then that would say that that is the phase in which the brain is sitting. So indeed, you can compare with the experiments, and you find out many things that are in common. For example, the grid orientations are discrete, the relative orientations of modules is preserved in individual animals across environments, the individual rats show consistent relative orientation differences consistent with the model, etc. There are some differences though, which is that the numbers are not bang on. The ratios are not exact, and the angles associated with each of these ratios are not exactly what you see. But we also left out things from this model, which is, for example, we left out the idea that in addition to this sort of self-motion, there's clearly anchoring to boundaries and landmarks that could happen, and we haven't included that so far in this study. Okay. So I'm going to conclude. I think, am I out of time, by the way? Just about. Because I've reached my conclusion, but I'm wondering how many minutes I have to talk still. Massimo, somebody.
You started a few minutes late. I think, you're kind of right around time.
Yeah, I was looking at my watch and I was wondering what time we actually start. Okay, so I'm going to conclude then by talking about this last set of things. So about what we left out. Right? Left out many things. One thing is thinking about borders, landmarks, error correction of mental maps, and things of this kind. So with regard to that, I've described a model by which these sort of mental maps of space, producing these kind of effectively 2-dimensional number systems for location in the world, can self-organize. But actually, any such dynamical system for self-organization is going to have a problem, which is that it accumulates errors in its path integrations. As you move around, this will be a little bit of error. And once you have it a little bit wrong, it doesn't go away. It stays there, right? Because you have no way of correcting it. And then as you go along, you'll get positional drift. You'll basically random walk it a little bit, right? So there'll be some diffusive drift that increases the error in position over time. So how could you correct this? Well, an obvious way of trying to correct this is by interaction with boundaries. The kind of thing that we said would probably also slightly correct what the dynamical model said about these hierarchy of grid modules and their ratios and so on. So it turns out, as I said, there are other things in the entorhinal cortex, like border cells, which chart out, map out little bits of the boundary. So then you could invoke other kinds of mechanisms. So for example, you could imagine that there are all of these grid modules at different kinds of periods. And let's suppose that there are border cells that are attached, which fire at different locations of the border. You can imagine invoking the entire mechanisms of learning in the brain using something called Hebbian plasticity. So Hebbian plasticity is a mechanism where if you've got two neurons that are connected and they fire at similar times, then they increase their synaptic strength. So this is a very simple way of self-organizing a system, somewhat different from what I was talking about before, but another way of self-organizing a system, where in here, you could have the border cells and the grid cells wired together if they share the same phase, right? So if a grid cell has a phase that would make it fire at a particular location in the border, then you could make the border cell and grid cell fire together. And then what would happen is that if the grid cell began to drift because it accumulates errors in its path integration and it reaches this border, you could have the border sort of be a bumper mechanism, right, to shift it back. So if you do that, in a model anyway, what you do is you predict a significant decrease in this drift or the error that's accumulating. So, if you make up a theory like this, that it's interaction with borders or boundaries that will sort of error correct your system and hopefully also adjust a little bit our predictions for the scale ratios and everything in the dynamical system, then you need to have some prediction that you can test it with. So here's a prediction. So if you believe that it's the case that border cells interact with grid cells to sort of error correct them so that if they drift in phase, that they're pulled back, here's a prediction you can make. So let's start off initially with a border cell attached here and a border cell attached here and some sort of grid cells that have, this is grid cell 1 that has this phase and grid cell here that has this phase. By this phases, I mean this little bumpy regions, the regions where the grid cell fires, and these are two different phases. So now I imagine Hebbian learning happens. So this means that this border cell becomes wired to this grid cell and this border cell gets wired to this grid cell. So now let's imagine what happens if you have stretched the room. If I stretch the room, then, actually, when you get to this end, you should be out of phase because, the room has gotten bigger. But because you've grown this plasticity, because you've grown this connection between this border cell and this grid cell, what it will do is when you hit the right wall, it'll pull the grid to the right because it thinks that it should error correct you. And whereas if you're on the left here, it'll pull you to the left. So this mechanism for error correction then makes a prediction that if you stretch or shrink the room, you will find that the grid shift to the left or to the right, right? That there'll be a shift, a dynamical shift, depending upon the last wall you touch. So that's a prediction of such a model. And indeed, you can check this. You can go to data, and then you can look at where the grid cells fire, and you can sort of condition them on touching the right wall, where's the left wall, where's the north wall, south wall. You can stretch the rooms. And what you find is in the data, there is a clear evidence of the predicted grid shifts, suggesting that this mechanism I just described does exist as a way of tethering the sort of self-organized map of the world to the actual boundaries that define the edge of the world that you're working with. So what are the next steps? So this is an area that's just starting, and there's just so much to do. So I clearly described the base level of understanding things about how these circuits, how the circuits should be organized in terms of an optimization principle, then how the circuits self-organize in terms of a dynamical principle, how the circuits then correct their errors in terms of a different dynamical principle. But I included, for example, the latter model, border cells that map out bits of the border. And I didn't tell you how the border cells self-organize. There are also head direction cells, which sort of tell you where you are pointing your head in the world. And I didn't tell you how that overall network works. So there's a lot of work ahead to sort of understand the full dynamical self-organization of that entire system. Most of this work has been done in very simplistic settings, square rooms, circular rooms, long tube-like rooms, etc. Whereas, there was a rat living out here in my garden during the recent snowstorms. It was burrowing under everything, and then when the snow melted, I went and took a look at the photographs of its burrows. It's this worm-like things with chambers and things going out all over the place and so on. So those are the kinds of complex naturalistic environments where animals actually live, and almost nothing has been done to talk about whether these mechanisms we're talking about work in those spaces. Very recently, in the last couple of years, it's been shown that the exact same mechanisms we were talking about today are also reused when people imagine abstract spaces and when animals have to navigate abstract spaces. So the same things are what we're doing to geometrize all manner of phenomena. So it'd be very, very interesting to think about that. Amongst other things, how do you map a given abstract space into the kinds of dynamical mechanisms we're talking about? Because that's how you produce the representation. Especially interesting is high-dimensional spaces. So, if the abstract space you're talking about is, k-dimensional, and you have this structure, right? I described how motion in 2 dimensions can get mapped to the structure, but the same structure must get used to imagine a higher dimensional space too. So how does that map work? There's something interesting to be done there. A very cool thing that people are thinking about lately is the distinction between the egocentric experience of the world and the allocentric map you have in the brain. Egocentric means relative to you, right? So your experience of the world is that thing's ahead of me. This is to the left of me. That is to the right of me, etc. The map you have in your head is allocentric. It's that way north versus that way west. So there's some transformation that occurs from the egocentric coordinate system into the allocentric coordinate system. Very little is known about the dynamical mechanisms and the anchoring that allows that to happen. So that's a very interesting thing to think about. On the other hand, having said that, we should consider Gugu Yimtir, Kaya Dilt, and Tayor, which I'm sure I have mispronounced, which are 3 indigenous cultures of Australia, where there is no word for left and right. You can't say it. You would tell somebody else, why don't you go into the ice cream store that's 10 feet to the north? You wouldn't say to the left because you can't. So it's clear that it's not necessary to have those relative components. So it's sort of interesting to ask how the sort of relativeness of much of our vocabulary actually interacts with the sort of absoluteness of the maps we're making the world. Anyway, I hope I've convinced you that this is an exciting and really fruitful source of really cool problems for theoretical physicists. I'm very glad I had the chance to think about base square root E numbers in 2 dimensions, and it would have never happened if I wasn't thinking about this problem. I'll stop there.
Excellent.
Can I ask a question?
Of course.
Should we raise hands in the reactions? Is that the way you can do it?
That's a way to do it.
Go for it.
You mean in the reactions or in chat?
Well, somebody should be the MC.
Reactions, I think, does it.
Okay, I'm gonna start calling on people. I think Massimo said, "Can I ask a question?"
Yeah. Okay. So the very simple. How many of these layers there are in typical?
Excellent question. You mean in terms of these modules? You mean, right?
Yes.
Yeah. So these are exceedingly difficult experiments to do. So they have managed to send the electrodes in through half of the structure and found about 5 modules. So the expectation is that there are about 10 modules. Yeah. So that's the answer. You could also have a discussion of why would you want 10 modules versus 56 or 3. And actually, in our analysis of this grid spacings of, if you look at you can do the following kinds of guesswork. So you can look at the native range of the typical rat. It's like it'll roam over some 50 meters. And you can ask how big a resolution does it need? And you could guess that the resolution it needs is about the body size. right? Because things that are much smaller than body size, well, you can't really resolve anyway. And so if you ask for the typical ratios of ranges that way, you need about 10 modules with a grid scaling of about square root of e in order to get that resolution. So it's not inconsistent. Well, I won't say that proves anything, but what it does say is it's not inconsistent with what one would expect from the behavior, from the ethology of these animals.
Thank you.
I think Neil had his hand up.
Thanks, Vijay. That was a really, really interesting talk. So I have sort of a maybe sort of crazy question, but maybe you were alluding to it on your second to last slide where you said that that some of these things are seen in humans in VR environments. So I was kind of curious about the application of this to humans, and I was starting to think about whether this is something, these sort of things are things that you could, if rather than just like randomly train, if you like actively wanted to do some skill building and you wanted somebody to rapidly build a map of a local environment. Imagine you're like a sporting team or something like that, and you want to train your athletes to very quickly be able to build a local map of the environment or something like that. I don't know. Are there things that this then instructs you that you could do to kind of like rapidly build up that sort of a map?
Let me answer this in various stages, going backward from the last thing to the first thing that you said. So the answer is, I'm very sure that this will instruct us, that these kinds of ideas will instruct us in ways of doing that kind of thing more efficiently, because there are absolutely more efficient exploration paths for gathering the necessary information to build a path. So if you just randomly wander around, you will not build a map as efficiently as you should. That analysis has not been done. So, but is something that actually I've been thinking about in collaboration with a colleague at the Technion, in which we are in the process of trying to build alternate mazes of deformable environments, and then try to make predictions about what kinds of navigation paths do what and so on. These are experiments with mice and rats as opposed to humans. However, you could definitely do it with humans. So for example, there is evidence from various kinds of, first of all, recordings from people who have okay, so before you do surgery for epilepsy, you insert electrodes in the brain to sort of figure out what's going on. You have to map the brain out. And so sometimes when you're doing that, the patient will say, go for it, I'm interested to find out something about the my spatial maps or something like this. And from that, there's some evidence for grid cells in humans. And there's also fMRI kind of data suggesting that there are grid cells and things like this in humans too, that they're parallel. I'm particularly fond of the piece of evidence alluded to in the little thing on the right, because it's completely non-invasive, and it's a thing you can do at home if you wished. You put on one of those virtual reality goggles and you make up a room. And you have to design this very carefully with fog and all kinds of stuff, because otherwise, people are very smart. They'll use every random cue to avoid you being able to test them. So what you do is you make a room, you proceed to sort of train them up as to the locations of different objects, right? And then what you do is you secretly spread the room in the fog, of course, so they don't know you've spread it. Otherwise, it's like this. A human will pick it up instantly that you've done something. And then you ask them, replace the object. And you will find that people shift where they put things at, depending upon which wall they were last at in exactly this kind of manner, suggesting this kind of boundary anchoring is happening. So on the one hand, it's like, well, duh, of course you're anchored to the boundary, what did you think, right? On the other hand, it's not as trivial as that sounds, right? It's easy for us to say. It's quite a different thing to say something about how the circuit, would produce that kind of anchoring. Because it's not like somebody goes in and says, you tell this person that you should be anchored this way. It's just a bunch of neurons doing whatever they're doing. So it's very interesting. And I am sure that there will be ways of thinking about how to train up more efficient ways of learning spaces out of this. I'd just like to mention that there are lots of, since ancient times, for all I know, those tricks are already known, because people have thought about this kind of thing a lot. Like, for example, one of the most famous tricks for keeping a long list of things in the memory is called the memory palace of Simonides. And the way this works is you take an enormous list that you want to remember, and you imagine a gigantic palace or something, and you take the objects one by one and you place them in your head. In your head, you place them. I put that on the shelf. Then I go to the coffee table. Then I go to the ledge. Then I walk to the other room, and you sort of say this to yourself, and you put it all down. You're going to remember it, right? Your memory is tied to space. So that's known because people have had a long time to think about how to remember stuff. So it wouldn't surprise me if there are tricks for learning how you learn a map of a space. I just don't know them myself.
Thank you. So I am the host, so I suppose I cannot raise hands, but I can see hands. Paul, Lauren, Shura, Alan, Mark, I have all hands up. So maybe alphabetically, Paul.
Okay, great. That was terrific talk. How many dimensions do we encode things in? And in particular, do we encode time a similar way?
Oh, these are two such great questions. Okay, so first of all, so the tests on terrestrial animals have been done in 2 dimensions. But it's obvious that we clearly do stuff like climb up, ladders and things like that. That hasn't been tested at all about how you deal with that 3rd dimension in the head of a mouse, a rat, or a person. There was one little thing done where there was a room and they put a divot in it, right? The sort of goes down. And then it seemed as though in that space, the mouse treated it in its head, the rat treated it in its head as still 2-dimensional, but sort of, how to say, it's like in general relativity, you re-coordinatize the space to sort of stretch it out. That's how it treated it in its head. In this. Super cool, right? Okay.
How about 4th dimension?
However, let's come to time in a second. However, there are things that move in 3 dimensions. Fish, bats, and there is a wonderful set of experiments being done at the Weizmann Institute and now also at Berkeley involving bats, where you can make these very lightweight electrode arrays that you mount on the bat's head and just set the bat free. And it flies these enormous distances, 10 miles, stuff like this, and they're able to track it through these gigantic tunnels. There is some information suggesting that there are 3-dimensional grid cells in the 3-dimensional representation. That's preliminary, because doing these experiments where you go deeper and deeper into the brain and sort of see these grids of different sizes and everything, that's exceedingly hard, even if you have an animal walking around. You can imagine what it's like in flight, because the brain sloshes a bit, and there's all kinds of issues with doing that. A further confounding factor is that although bats fly in 3 dimensions, they live quasi 2-dimensional lives, because they spend a lot of time kind of walking on the surface of the cave and kind of hanging around near surfaces and stuff like that. And then every once in a while they take these long flights. So it's more murky, and the story on this is still being told. It's just beginning as of a couple of years ago. Very, very interesting. I think fish would be a target too, except that how you record from those things when they're underwater, I don't know. But anyway, but there must be a way. So that's interesting. Time is really interesting. So this is work coming out of BU, and there are time cells in the brain. So how there's the somewhat strangely named time crystals we talk about, which is simply periodic things in time. So indeed, there are cells that sort of keep time. There are various kinds of rhythms. There are things that beat, that will fire at specific, just like a grid in space. A grid in space, periodic space. A grid in time is periodic in time. So there's things that keep time in that manner, and there's all kinds of interesting scaling relationships and things like that about those. I do not know that literature very well. That is a target for something I would like to think about in due course. But there is definitely, they're in the same structures. They're very important because, for example, if you're planning a trajectory, why do you need to know where you are? It's because you need to know where you want to go, and you want to know how to get there. So that involves time, right? Even just imagining the path, which animals do, by the way. If you look at these structures and they're asleep, right, you can see them replay paths in this. You'll see the grids move. You'll see the place cell activation as if it's walking down a path, right? You can see that, right? And so it's almost certain, I think, that you imagine those paths. But for that, you need a notion of time, because you need to sequence it. So there are time cells, and how they interact with everything else is not as clearly known as one would like, but that'll come.
But it is hierarchical. It is different scales.
We don't exactly, right? There are different scales. That's known. That is known. There's the hierarchies of these things, but we don't know. We don't know that these things with the kind of precision that I would like to be able to say, look, I'd like to give the same narrative, right? That suppose you want to know where you are in time with a certain precision, and I want to know this over a day. Then how should I structure the thing, right? You'd like to be able to answer questions like that, but I don't know have to do that. But it's definitely coming, right? I mean, sometimes I actually think that grid cells are actually time cells in a peculiar way. Let me explain what I mean. So grid cells respond when you are in a lattice of locations in the world. But how do you probe the lattice of locations? You walk around them. So a grid cell, as you walk around them, will fire periodically if you're walking in a straight line. So it turns space into a periodicity in time. So there's some very interesting connection there, right? About temporal periodicity in the firing of these things, and the nature of the trajectories, and there's some interaction there that I do not understand, but that is there.
Thanks.
You, Lauren?
Hi, thank you so much. This was incredibly interesting. And I was just wondering one thing about kind of the like, if you're doing an experiment on an animal and they're moving through a space and they learn the space and they develop the grid cell like orientation for it, is there like any way that those same cells can activate when that animal is watching another animal doing those same movements and interacting in that same way?
That hasn't been tested, but almost certainly, in my opinion, the answer will be yes for some animals. So some animals, definitely primates, these experiments have been done with, some experiments have been done with primates, not with space, but with other kinds of structures in the brain. So you can have a primate, another primate, and they're friends, and this primate is doing some tasks like moving a lever or something. And if you look at the first primate and you record from its brain, you can see, or sometimes it's fMRI, you see what's being activated. You can see that it's mirroring. You can do that. I suspect not all animals will be able to do that. That's probably a pretty sophisticated capacity to have. But some animals definitely can. Imagine, they must be, all kinds of animals. My parents described this thing where they were sort of walking down the road and they saw some bird, just hit something and be sort of stunned and lying on the road. And then they saw two birds of some other species, some other bird, whatever it was, come down and kind of look perturbed and pick up the first bird by the wings and put it on the side of the road and leave, right? That requires a level of empathy that's kind of amazing. And so you have to be able to imagine in your head the condition of the other animal. And so imagining locations in space seems like a minimal version of this. Also, by the way, many animals are social. So if you want to learn from what the other animal does, oh, look, it walked over there, and when it got to there, it got the M&M, right? If you want to learn that, you need to be able to make the mental map based on the other person's motion, right? So, and animals do that. A mouse can watch another mouse and figure out what to do based upon the experiences of the first mouse. So it must be the case, but it's not known. Another cool experiment waiting to be done.
Cool. Thank you.
So Shura, we're going down the list.
Hi, haven't seen you in a bit.
Yes, how are you, Vijay? Thank you for very nice talk. And by the way, I think we should all thank the students for inviting Vijay.
Yeah.
So I have a couple of questions to you, Vijay, in continuation of actually Paul Chaikin's question. So what happens if I take a baby mouse and from the very beginning, I place it under some computer supervision in a virtual world, virtual reality, which is actually 4 or 5 or 6 dimensional.
Mm-hmm.
Would it learn to live in D dimension, and would it produce, according to your prediction, 4th or 5th or whatever root of E spacing?
Wonderful question. First of all, I'd like to point out that the 6th root of E and the 5th root of E are both, unfortunately, very close to 1. So probably within the experimental error bars, we can't tell the difference. I think it's a tragedy. But anyway, having said that, you can certainly do 1, 2, and 3 dimensions, and they're different. You'll get different numbers. So this is an experiment I dream of. Now, being a theorist, Shura, there is this whole art to experimentalist management. you have to become friends with them. You got to feed them candy. You have to sort of give them little snippets that they can use in their papers. It'll make them happy. You write a few papers with them, then you gain their confidence. And then one day,
Vijay, I can tell you the trick is you have to place them in virtual reality, which is 4-dimensional. Then they will do what you want.
yes, probably. So
I'm so glad this is being recorded.
yes. So actually, having said all that, I do have a friend with whom I am working on these kinds of things. So these kinds of experiments. So let's review the history of these kinds of sort of modified reality experiments. So the early experiments of these kinds were sensory deprivation experiments, right? What you do is you take kittens or mice or whatever it is you have, and you raise them in an environment after eye opening, which have no vertical lines, none, right?
Okay.
And then what happens is you examine their visual cortices, and the cells that should be there, the simple cells and complex cells that respond to bars, are just absent. The brain is just modified by the experience of the young animal. This is why, by the way, you should not sensorily deprive your children, right? They should have all the sensory stimulation they want. So what you're describing is a much more sophisticated version of that, where you don't do this willy-nilly. I'll hit it with a hammer, right? There's a nothing. I'll give it a modified number of dimensions. There's a stepping stone to that, right? The first thing to ask is it could happen that these circuits are really so adapted to living in our world that they don't do this well if you take a young animal of that kind. So a stepping stone to this is as follows. So here is another simpler thing, okay? So it'll seem initially unrelated, and then I'll tell you how it's related. So consider the perception of textures by humans, okay? So, we're able to detect textures, say this texture is different from this texture. By texture, I just mean correlated patterns of light, right? Black and white spots and what have you, with different kinds of correlations. The structure of correlations defines the texture. So I have a series of papers with Jonathan Victor, who's a professor at Cornell Weill in New York, basically demonstrating the following thing. So you can basically define these classes of artificial textures. And then you can do an experiment where you place these artificial textures on white noise background, flash it very quickly. You have 100 milliseconds, boom, boom, boom. And you ask people, well, was something there or not? Right? Just detect it. And you find very interesting patterns in which you have two different textures, let's say, which are as statistically deviant from white noise. And one is clearly visible to you, and one is essentially invisible. It's just really hard to tell, even though it's just really different from white noise and by statistical measures. So you can ask, why would that be the case? And of course, a natural hypothesis would be this has something to do with the textural statistics of natural scenes, right? That basically some of these things are useful to detect and some aren't. So we have a series of papers which started with collecting an image database from a baboon habitat in Botswana, and there's this long sequence of things that was done, in which we then found a way to piece apart natural images into their textual content, and then showed, indeed, this idea that would be natural is precisely realized. Okay, but that is a post-diction, right? There's a certain thing that happens, and then we showed that you could explain it this way. A prediction would be the experiment you suggested, right? At eye opening, you graze the animal in a modified textural environment, and then you ask, are the things it can detect changed from what it would have otherwise detected? And in fact, that experiment is being done right now with my colleague Davide Zoccoli, who's in Italy, at CISA. And we have now established, or rather he has now established, that with the textures we gave him, that rats can do this task. You're not allowed, rightly, to do this with human babies. So,
of course, yes.
Right, you would do this with rats. So the first thing you have to establish that rats can learn to do this kind of thing, right? Because you can't really talk to them. So we've now established that they can do it. And so we have to modify, the visual resolution, all kinds of stuff. So that's working. So the next step is to raise these things in modified environments. So my Davide has also been talking with people who raise chicks. And interestingly, this has also been somebody who works on human babies has also been talking to him. I'm not sure exactly what they're going to do, but I think there will be an experiment there. Once that's in place, and we show that we can do this raising in alternate environments and everything, we can get more ambitious. And I would love to try what you suggested. So,
I see.
Couple
Okay.
of years down the line.
Thank you. Massimo, can I ask one more? Massimo, you're muted.
We have other three people, but go ahead. I think that people are interested in the discussion.
My other question is the following. Apart from the model of space as a place where I live, where I am a point in some sense, there is another problem of space, which is the spatial relations between parts of my body.
Mm-hmm.
So is this done in some similar ways? Is done by the same part of the brain, or is it completely different thing?
So I know less about this, so I will not be able to give you a precise answer. It may be, it's likely, this part of the brain is being used partly for that, but there are actually other parts of the brain which are specifically tasked with coordinating the different bits of you. So, coordinating sensory information, coordinating body plan information, things like this. So, for example, I mean, you need this all the time, and you need it in a
Of course,
different.
yeah. If I want to scratch my nose, then sure, of course.
Yeah, exactly. You got to move your head and you have to move your finger, right? I mean, so this requires a massive degree of coordination. There's entire systems dedicated to this. So how exactly the coding of all the locations of everything there is done and everything, there is work on that, but I don't know it well enough to make a meaningful comment.
Thank you, Vijay.
Thank you. So Alan is next.
Hi, I very much enjoyed your talk. I'm thinking about dynamic range. So if I'm thinking about a flea in a football field, either I would think I need very many layers, or I have a coarse view of it, then I take a finer view and a finer view. And I would imagine that you could even do an experiment where you have people imagine positions of things and see if you zoom right down to something or if you do it in steps. So I've been trying to imagine that now for the last few minutes, and I feel like if I, in my imagination, position a fly somewhere in the field, I can only do it to some resolution, and then I have to reshape my map and go again. So any thoughts about that?
Yes, so there are two classes of thoughts about that. So one class of thoughts about that has to do with something I actually don't know that much about, but clearly sometimes you want to imagine a space in a coarse-grained way, and sometimes you want to use the same map and focus it in some narrow region. I don't think that much has been done about that at all. So we can definitely speculate that you use the same structure and then sort of just basically scale it to the space. The reason why that's possible is because the inputs, you remember I told you about how there's a neural structure with cells responding, and then there's real space, and there's a sort of isomorphism between them that's produced by putting up some sort of velocity input, right? So that's how you sort of connect them, you tie them together. So there's a gain in that process. So there's some gain variable associated with how much current comes in when something comes in, that kind of thing. By tweaking that gain, you could definitely scale the thing up, scale the whole thing up and make it coarser, if you like, but covering a wider region or smaller. How and whether that happens is not really known because there are no experiments on that. It would seem absolutely natural that when you're imagining big spaces versus small spaces, you use that. The experiments haven't been done for multiple reasons. You can't tell a mouse or a rat to do that, right? You can tell a human to do that. You can't tell anybody else to do that, right? But presumably we do, and maybe there'll be someday a way of getting the information about that kind of rescaling. The other reason it hasn't been done is there are almost no experiments in enclosures larger than 2 meters squared. I'm aware of one experiment in a 10-meter long track done at the Geneva Research Campus of the Howard Hughes Medical of the HHMI, and these bat experiments. Everything else is in small enclosures. So this is an excellent question, because real animal behavior involves this kind of going over multiple distances. And when we navigate, it's very clear when we think about our maps inside the head. If I think about how I get from here to Philadelphia, I do not think of the street level map first, right? I'm thinking I need to get from here to there, and then I think, oh great, now I'm here, and I need to think about how to get from this street to that street. There's this sort of multi-scale thing that's being done on a regular basis. We must be using mechanisms like that, but I can't swear to them. Another thing that is known to happen is something called remapping. So suppose you make a room, you connect it by tunnel to another room, to a third room, you make a sort of chamber-like environment of this kind. So what is definitely known is if you learn these rooms, as you walk from room to room, the map reloads, a different map reloads in every space, right? Different place cells, different grid cells are attached to different places. Certain properties will remain invariant, like the relative orientation of grids can remain invariant between these places, but you will get a remapping that occurs from place to place. So another hypothesis you might make is the way you do the local, at least the local navigation, is that you maybe one could test this with the bats, that you have one grid here, then you move for a bit, then you reload another grid, and then there's the transition function that sort of puts these things together, and you keep going like that, right? It's like making a manifold out of tangent spaces, that kind of thing. So you make a little grid, another grid, another grid, another grid, and you piece those together. That doesn't answer the question of imagining the whole scale versus the small scale, right? And I think something like what you said seems entirely reasonable, right? And would be a very reasonable hypothesis.
Great, thank you very much.
It's Mark or David. Maybe.
So,
I think Mark was before me.
Oh, okay. Yeah, I was following this list. So, Mark.
So I have now two questions because you answered one of them. I'll be fast. But so the same place cells represent the different places as you move in environments. And so my question.
Let me just correct one thing. Today I want to talk about grid cells.
Right, same grid cells, right? So I guess one question.
Grid cells do a different thing.
Right. So I have one question about, so you can encode something efficiently, but then you have to read it out.
Yeah.
So do you have any idea how the readout is done efficiently?
Excellent question. Okay, so this is the topic that launched 1,000 papers, right? So let's start speculating on what you might do. Of course, the full answer is it's not entirely known, but here are some answers to that. So the entorhinal cortex, where these grid cells exist, was originally discovered as a place where these grid cells are because it is a primary input. It is a primary input to the hippocampus. And in the hippocampus, there are things called place cells, which I alluded to at various points. Place cells have the property that if I look at a particular place cell, at least in a small environment, I will find that it responds in one location in that space. It's more like the sort of place encoding that I was talking about earlier, okay? Now that's been muddied a little bit because it turns out that if you make the environment much bigger, then it'll turn out that these place cells actually respond in 2 places or kind of 2.5 places, and sometimes they change where they respond to. So inevitably, the picture is becoming a little bit murkier, okay? But to a first approximation, they respond in some location. The A theory of how place cells are built is that they come from grid cells. That you take the grid and you project the grid cells into the hippocampus, and then you have some kind of plasticity mechanism so that you gather together synapses between grid cells which have the same phase. So if you grab grid cells that have the same phase, then they will add up sort of coherently. And what's more, because you've got a big period, smaller period, smaller period, smaller period, perhaps by the mechanism that I was describing today, you will find one response field within the environment. And then you have a place field, right? So the readout was one idea for the readout was like that, okay? There are some murky things about this. Well, I noted a couple of things already, that experimentally now it's a little bit murkier. There are multiple place fields and everything. On the other hand, that might actually be a good sign. Because, if I take a whole bunch of grid fields and I don't do this kind of plasticity that I talked about, collect together the ones that have the same phase, and instead sum them randomly, right? Then mostly because they're out of phase, they'll cancel, if you like, kind of, sort of. But some of them will add up coherently, but you'll have some statistics of secondary and tertiary sort of lobes appearing. And so there's some suggestions people are making that they see those lobes, and those lobes are actually coming from this mechanism of summing things up. Okay. Now we'll make the problem a little bit murkier, having said that the place cells are the readout of the grid cells. Turns out that developmentally, the place cells appear first. So if you look at infant mice, you'll find that they have rudimentary place fields, but they can't make grid cells, which then suggests that, so you have a chicken and egg thing. You need place cells to make grid cells. Well, then how do grid cells make place cells? But of course, these things could be mutually consistent because infant mice don't move much, so maybe they make rudimentary place fields based on interactions with the mother and the wall and little things like that, and somehow this kind of then seeds the whole process, and then the grid cells form, and then they really make place fields. Many things are possible here, right? So another thing that makes things murky about the place cells being the readout of the grid cells is, here's a kooky thing about them, about which I have a paper that's been sitting on my desk for 7 years. I really have to write this thing and get it out. So here's a murky thing. The murky thing is that it turns out, if I want to know where I am and I have a place field that responds here, I'm done, I'm here. But it turns out that in the hippocampus, you'll find small place fields and bigger place fields and bigger place fields. So the same location is multiply encoded by small place fields and big place fields. By the way, this may have some bearing on Alan's question earlier about coarse scale versus small scale representations of the world, right? So you basically, the same location is multiply represented at multiple scales. So then you can ask yourself the question, well, how am I supposed to do that, right? If I summed up grid fields with the same phase to get this little place field, how do I get this thing with the bigger phase? Now, one potential answer to that is that what you could do is, well, we said there were these 10 modules. Let's make the little ones, little place fields out of these modules, and the bigger ones out of the bigger modules, and things like that. So that's another possibility. So I'm now sort of just spewing n different possible hypotheses. That's the state of it.
David was nice enough to let me go, so I should let him.
Okay, let's see. I have an experimental question. Now, this is sort of dangerous asking a theorist about experiment. And so hopefully you've made good enough friends and fed your experimentalist friends enough that they let you in on the secrets. So it's, the magnetic resonance, it doesn't have such good resolution. So I imagine to get good resolution, that's all comes from poking these electrodes inside of
what doesn't have good resolution?
Functional NMR.
Yes.
So the good resolution on a sort of single neuron level comes from putting little electrodes into animals' brains. There's lots of questions I have just about the experiments. First of all, how densely can they sample? Can you put enough electrodes in that you sample individual neighboring electrodes? That sounds kind of hard.
Okay, so let's, there'll be...
And then there's lots of questions. To what extent do you disturb them? Hopefully you don't puncture the neurons. And how the hell do you know where you are?
Yes.
You're going through, you're seeing all these spikes and so forth. How do you know where you are? Experimentally, it just seems like a morass, and I wonder how people actually get clean data and what the spatial resolution of those data are in terms of. And then the neurons, as you pointed out, they have all these axons coming out. What does it mean a neuron fires? My head's swimming.
So let me I'll try to remember. There were about 4 questions there. I'll try to answer them, and if I don't, then you should ask me. So when I started working in this field, I was a postdoc doing string theory, and I spent in the evenings I would moonlight in a neuroscience lab, sitting with the experimentalists there and looking very carefully through exactly what they did, what the experiments were like, what the data was like, and getting them to tell me all the actual problems, because you can't interpret this kind of data without actually knowing what what's going on under the hood. So every question you asked is important, right? So let me try to take every single one of them one by one. So first, concerning the resolution, that was the first question you asked. So that has been a process of historical change, a little bit like what happens with microchips getting denser. So when I first started looking at neural data, so originally people would poke in one electrode, and you could do one of 2 things. You either put it in and you kind of hope to pick something up, or you patch onto a neuron, and then you have a very tight seal with that neuron, and you get beautiful data from one neuron. You either poke a hole in it and then suck it in so that it's sort of held, so that it's not leaking, right? You poke a hole and you go, and then it seals, and then you can record from it, or you sort of patch outside it, and things like that. So those were called patch clamp experiments. Beautiful data, but you'll get one neuron at a time. And evolution in this was the invention of things like the multi-electrode array. So the early arrays would have like 60 electrodes, fairly far spaced apart, because this was sort of very difficult technology at the time, but they were flat. So what you would do is you could do things like the retina, flat structure. So you'd put the retina on top. I have done these experiments myself, so I know just how difficult they are actually to get everything to work out properly. Because you put them in, you get tiny signals because nothing's poking in anywhere. You're just sort of squishing it down. You got to keep the thing alive. And you might have 60 electrodes, but your problem is that every electrode gets signals from many neurons, and every neuron reports to many electrodes. So then you have to solve this multiplexing problem. It's called the spike sorting problem, and which is a very interesting inference problem to solve and challenging. So in the data that I used to get my hands on, there'd be 60 electrodes, and it was a good day when you got 4 neurons out of it. Okay, so that's not very dense, right? There's like 100,000 neurons in the guinea pig retina, output neurons, and you're getting 4 of them. So that's no good from a certain point of view. So that technology has been improving steadily. So for example, there are CMOS technologies, this, that, and the other, and so the flat arrays have been getting denser. Even better, there have been a whole array of new kinds of insertable probes. So various new kinds of electrodes that'll give you, in the last decade, the things that give you 10 neurons at a time, 50 neurons at a time, things that had a shaft and have multiple electrodes sticking out at the same time in multiple directions, so you could get many electrodes at the same time. The first thing was the tetrode, which had 4 electrodes, and then it went to this sort of multiple electrodes of this kind. There is a new kind of probe just out called the neuropixels probe, in which people are reportedly getting hundreds of neurons. Right? I have not gotten my hands on any data of this kind. My collaborator at the Technion has just got a neuropixels probe, and I am very, very excited to see for recordings of the hippocampus and the entorhinal cortex. So I'm very, very excited to see what will happen. The experiments I talked about were using tetrodes of different kinds, and with long shafts going in, or maybe there were more than tetrodes, maybe there were like 8 electrodes, 16 electrodes. So the typical experiment would get a few neurons, not very many. That's part of the reason why these are so arduous and sort of heroic feats by the experimentalists. They face many problems. When you push this thing in, yes, you run the danger of poking into the neurons and damaging them. You don't want to damage the brain area. So you have to be very careful going. You're still going to probably damage something sometimes, right? So the electrodes are somewhat flexible, so they're supposed to kind of bend out of the way, but who can guarantee that, and how can you be sure, right? So that is one whole class of issues there. Can you guarantee you're getting neighboring neurons? No, you can't with many of these things, because it depends upon where the signals are coming from. And it would be great if you could triangulate. I don't see why you couldn't, right? You could have something going in here, another thing going in there, another thing going in there, and then you triangulate using the fact that, it's signal propagation, right? But there's no electrode of that kind that people have used. I wrote a spike sorting algorithm for square arrays. This is with Phil Nelson. I've written an algorithm with square arrays of electrodes on which you could put the retina, because there you could use geometry. If a neuron is here, I know I can try to back out the dissipation of the signal across the electrode array, and then back from out of that. But you need to know enough geometry there, or have enough density of the probe. So those are all issues that exist. I didn't address everything you said. You said, where does the signal come from? So you said one thing that was not quite right, which is you said there are so many axons. There aren't. There's one axon. The cell body has one axon, and the axons are the long wire out, and then branches at the end. The little bits and at the axon tips are going to be small. So the bulk of the signal you're getting is probably coming from the cell body, right? Because that's where the action potential is generated. So that's a pretty chunky amount of membrane. So that's what you're really trying to measure, because the spike is produced in the cell body and then propagates down. There are other kinds of probes you'll be happy to hear. So there's a thing called a miniscope, which is a tiny little sort of thing that you can put in. That's like a little microscope that resolves things at high resolutions inside the brain. And there are new optical tools that you can use that with. So there are many kinds of now electrically active dyes, and also ways of genetically modifying neurons, right? Inject viruses that put little bits of something into the genome, and that produces some protein, and that protein glows when the potential goes over something, that kind of thing. So then if you can do that, then you can do imaging using that kind of technique. Another kind of technique is calcium imaging, right? You can image calcium. There are dyes that react to calcium. Typically, those kinds of optical techniques have a lower time resolution, but they're also in some ways less invasive because you're not poking stuff all over the place. So there's also calcium data of that kind instead of optical imaging data of that kind. So that's also very valuable for this kind of work. And that's another set. I actually have never personally worked with one of those data sets, but I think again one of my collaborators is gathering one, and I hope I will have a chance to see what that does. Very, very important technique. There's even more magic that I made a bit of fun of how you have to manipulate your experimentalist friend doing the experiment you want them to do, but they do magic, right? And part of the reason that they're reluctant is it takes so much effort to set the damn things up and do these experiments that you have to be kind of sure that something useful is going to come out, not just some theorist shooting their mouth off. So another kind of very interesting experiment involves an activity-dependent thing. So what you want to do is, suppose you want to find out if something is connected to something. Well, what you'd like to do is if this triggers and then activates that neuron, you'd like it to keep a barcode, or you'd like it to keep glow or something to tell you it was activated. And then later, you'd like to be able to go and activate the neuron that was activated before with a laser, let's say, right? Optogenetically. So there are now technologies for doing that kind of thing.
That sounds like a lot of fun. That sounds interesting.
Isn't it amazing?
Yeah.
I mean, these things are just completely amazing, right? I mean, they're like, they're indistinguishable from magic. Right? I mean, they're just really amazing techniques.
Okay, thank you very much. That got me a little ways along the way to answers. It's obviously pretty complex.
Thank you, Vijay. So let's thank you again, so that people that have to leave can leave, and then we can continue also with our discussion. It seems very, very nice. So thank you for the
Thank you very much for having me.
generosity with your time.